Um, yeah. Yep. So welcome to the July ARC2 Open Science meeting. Again, it's been a while since we had gotten a chance to get together. The last time we met was around in March. So there's quite a bit of update to give. So for the agenda today, I'm going to just give a brief introduction just to kind of bring people back on track with where we were and what we are trying to accomplish. But what's most exciting is that we're going to be getting some in vivo updates from Angle and Diana in their model. So we're all excited to hear about that. Then we're going to get some updates on the chemistry efforts from Charles River by Sue Graham. And then we wanted to give you some updates on some of the meetings we had over the past 2 months. We had an SAB meeting and we had a CITIP meeting. It's important to introduce the team to one of the new leads for the project, Dr. Peter Ho, who has now come on board. And so he has established an SAB team and we actually had a very, very nice meeting. So he'll provide you with an update in terms of some of the outcomes and feedback from that meeting. And I'll also provide some updates on the CTIP feedback that we had recently. And then in the end, there's going to be a chance for any burning questions or discussion. Okay, so just to remind people, since the last set of actions we had since March. So again, like I mentioned, we were doing this DIPG efficacy study, which was an essential part to the completing the actual CT project at OICR. So what we had ongoing was 2009 was being tested in the Becher model by Diana and Chris Jones. Angel also looked at 2163 in his orthotopic model. And then we also had Nader, who was actually looking at some sub-Q versions of these. At the same time, we're still trying to understand some of the biology in these lines. So Alex and his team continue to do a little bit of effort in the ALK2 activity for the lines. And then we had the backup effort from Charles River along with the POTAC effort. So these were some of the things. Some of these are still ongoing. Unfortunately, with the COVID situation, we had to ramp down a bit, but slowly, things are coming back on board. Just again to remind the team, what we have done is basically we had one lead series, the pyridine series, which we have advanced, and this was a sort of a nice snapshot summary of the lead optimization of the pyridine series. But Charles River has been working diligently on the backup efforts from the blueprint series. But now you're going to see from Sukram's update, they might start getting back involved based on some of the feedback. We have been getting back into the pyridine series, looking for more potent compounds. But all in all, it looks like from our list of compounds that we had 5 lead candidates. We have now whittled it down to 2009 and 2163 as the candidates basically to test in vivo for proof of principle. And this is just to show you what the structures of those are, just in case anyone forgot. So these are the 2 compounds we are evaluating right now. With regards to competition, again, there are no selective molecules for ALK2 in the clinic. I mean, there's an ALK2 inhibitor advocated by Tolera, but we have evidence that that may not be very selective. They're probably hitting a lot of other kinases. So we still stand a good chance competitively. Most of the other companies are going after FOP, which is a non-CNS indication. But it's interesting, just this morning actually, I just saw this patent publication just came out from Insight this morning. And I thought I'll just share it with the group. Here's a summary. But it's interesting they're starting to use some of the features from our pharmacophore. And so the claim has an IC50 of about 1 nanomole. And there's a patent number. I didn't get a chance to pull it yet. So just keep an eye on that. That just got published this morning, hot off the press. So the competition is beefing up. Okay, so that's it for background and intro. If there aren't any other questions, I'm going to probably hand it over to Angel to provide his update. Any questions?
I need you to allow me to share my
Stop,
screen.
stop share. Yep, there we go.
Should I do it?
Yep. So I've stopped sharing, so you should be able to share now.
Okay. Can you see it?
Yes, yep. So, yep, there we go.
Okay.
So go to full. You could go to full screen. Yeah.
Yeah. That's it. So we have been testing this M4K 2163 here in Barcelona. The model we have used is the DIPG number 7 with the ACVR1 mutation R206H. We know this model is very good because it has a 100% take rate with a median survival of 70 to 100 days in previous experiments. And it is very diffuse, infiltrative, we all know it. Leire Balaguer has been very involved here, so I really want to appreciate her role. So here we perform in vitro studies just to test the drugs. This 2163 looks really potent with an IC50 less than 0.1 micromolar, which is really potent. Around 89 nanomolar in the DIPG7, much potent than the other one. This study was started 4 months ago, March 15, 2020. The previous day to the lockdown, the same day of the lockdown, by the way. I did all this, we did the surgeries here. And we started treatments around 35 days later. The design of the study was 4 groups. We have a vehicle group, 100 milligram per kilo group for 4 weeks. Half dose, 50 milligram per kilo, 4 weeks, oral. And then we continued 8 weeks with group number 4 with 100 milligram per kilo. So we collected three samples from each group, 1 through 2 and 3, not from the group 4, because group 4 and group 2 are the same until week 4. So at week 4, we studied what's going on in the brains. So we sacrificed 3 mice from groups 1, 2, and 3, okay? And with this, we did some kind of experiments here, or analysis. And the remaining mice, we observed them for survival. So we have 9 to 10 mice from groups 1, 2, and 3, untreated after these 4 weeks, and 10 mice from group 4. They continued receiving this treatment 4 more weeks. So 100 milligram per kilo for 8 weeks. The endpoint criteria is either moribund animal, you can notice very well, or 20% weight loss due to the disease. So if the animal is sick and weight this amount of weight and lose this amount of weight, that's considered the endpoint. But if you have a severe or harsh aggressive treatment, such as 100 milligram per kilo for 8 weeks, we don't consider this criteria that much because the animals lose weight more than 20. So we look more at the moribund status. Okay, is that clear? Because otherwise we lose the experiment.
Mm-hmm.
So these are the samples we collected 2 hours after the last dose at week 4 of treatment. So animals were treated 5 days on, 2 off during 4 weeks, and we collected the samples. We don't see any evidence of less tumor load in the brains of animals treated with 50 milligram per kilo or 100 milligram per kilo. What we analyze here is by digital droplet PCR, the amount of ACVR1 mutant DNA. This is a method to measure the tumor load in these animals. So this is what we see at the end of 4-week treatment. Okay, next. We stained the human cells in the control animals, and we don't see any difference between the density of these cells. You can check here for the control animals, here for the 100 milligram per kilo animals, and here for the 50 milligram per kilo animals. We cannot notice any difference in the tumor load staining the human nuclei. And we cannot see any difference neither staining for the proliferating cells, Ki67 here or here. So this looks like there is no much activity. We continued all the study measuring, weighing the animals almost daily. And we collected all this data to do the survival curves. This is the result. We have the black curve is the control animals. They start to die earlier than the treatment groups with this criteria of 20% weight loss from the maximum weight. But this difference is not significant compared to the blue and red, which are 100 milligram per kilo and 50 milligram per kilo. Survivals, median survivals are the same, 92 days for the control, 94 days for the 100 milligram per kilo, and 94 for the 50 milligram per kilo. However, in the group 4, the animals look better. They lose weight due to the treatment, but they look better in the health condition. So that's why they are not considered moribund animal. So far, median survival is 121 days, still for animals alive, and they look better, much better than the previous, than all the animals that died so far. And this is a little significant. If we compare all the 4 groups together, I'm not sure this is going to be significant, applying the Bonferroni correction, but so far it looks okay. So believe me, all these animals will die, but they look to have benefit of this treatment, 100 milligram per kilo maintained for 8 weeks, okay? This is what we have so far. And what we are going to do, what we can do is to ship all these samples to you. We have collected samples from all these mice. We have plasmas here, brain tumors. So this is ready to ship to you guys and to continue the study. Maybe you can see other parameters related to drug activity that we cannot see regarding tumor load. So this is something we would like to check. And this is all I have to share for today.
Okay, thank you. Interesting. Out of the first update. Have any burning questions for Angel?
Angel, it's Diana
Hello.
here.
Hello, Diana.
I was wondering about, the 100 mg per kilos, the 8-week one, those animals that are just dying at the end of the treatment. Do you think, because then you did that long survival going, you think those that died are due to toxicity or the tumor itself?
No, we don't think so. We think they are full of tumor. Yeah.
Okay, cool. Thank you.
Yeah, the treatment was toxic, but not super toxic. So they lose weight, but they didn't die due to treatment, I think.
Okay, cool.
Thank you.
When you say they lost weight in the in treatment group 4, how much weight loss were they having, and what did they look like? I know this wasn't a tox study, but
like
how did they look?
Like 20%. Oh, and this is the curves. These are the curves.
Okay.
So you can see here, this is group 4 versus group 1. So group 4 drops the weight faster than group 1, even though group 1 is the control. But then they keep the weight like more stable than the group 1. And here, some of the group 1 look very moribund, and we have to take the animals off of the study. And these ones, for example, these 4, they look healthy. Not healthy, but they are in better condition than the others. So this is what we have. Of course, we need to work on this, how to justify this design of the study, because we don't want to lose the study being too strict with this 20% criteria. If the drug is doing the 20%, then we don't see the real benefit. So we need to think of it and provide a good justification to say these animals are better, in better condition, and we ensure they are in better condition. We have these animals are very stable in their weight. This, you see, these, for example, this one here, this green one here, is dropping like, you see, very sharply. And this animal is, even though the weight was okay here, it is dead here, right? Because we have to have the endpoint here. However, these ones are still stable. This is what we have.
And other than weight loss, again, I know this wasn't a talk study, so you weren't taking out, but how were they active? Were they,
Yes.
Yeah, they were, so okay.
Yes, they are, they look better, more active. They don't look sick. They don't lose the balance as they in such a bad condition as the control animals.
Mm-hmm.
So we are pretty sure this is true. However, I think we can improve. We need to think about it. Yeah.
And what would be your thoughts about that improvement? Just
Exactly.
off the top of,
Yeah, I think maybe we started treatment too early. Maybe we wanted to start treatment instead of 35 days, maybe closer to 50. I don't know. We think we need to think about it and discuss together. I would like to repeat the study, of course, using this 8 weeks treatment. Maybe get rid of or keep the 50 milligram per kilo also 8 weeks to compare and get rid of this group. That's one option we can do.
Can you go back to the curves, the
sure.
Success curves for a second? Just the 100 mg per kg. Yeah, I was just thinking that the 100 mg per kg and the 50 mg per kg at the beginning don't look that different.
They don't. I would say these three groups, the difference is just random. I don't think they are different.
Yeah, because you would have thought the 4-week
actually
and the 8-week group should be the same.
Blue and green should be the same till here, right?
So how many mice are we talking is the difference between that during the...
Say again.
Oh, I'm just trying to think of the number of mice, the death of mice. It's probably, we're looking at 2 mice, aren't we? Is the difference.
Yeah, yes. Here.
This is Peter. Can I just follow up on, I think, the point you're trying to make, which is, you know, in the period between 60 to whatever, 80, 90 days, right? At least initially, there doesn't seem too much of a drop. But, you know, the point being that for groups 1 and 2, you know, deaths, right, really don't start occurring until well after treatment is stopped, right, in the pink period. And likewise for the 100 milligram, for the 100 milligram for 8 weeks treatment. Once again, you know, the deaths don't start to be observed till after treatment is stopped. So it does argue that if there's not much difference between the 50 and the 100 early on, that trying 50, as Engel had mentioned, for a longer period, right, might be less toxic because there are fewer animals that drop below 20% weight loss in that group, and you know, you're just sort of extending out the period of treatment and likely pushing out the survival curve.
That's like a very good point, Peter. I agree.
I'm sorry. Can you go back to the weight loss? That's a great point, Pierre. I'm just trying to see on the 50 versus the control what the weight loss was looking like. How healthy did those look? So that would have been group 2. No, that's group 3. Group 3 and group 1. So group 3's weight loss.
I mean, there's certainly great weight loss in group 3, and it seems, you know, somewhat comparable to group 1. But if you sort of draw a line at 20% or minus 20%, right, there are fewer animals, you know, that drop below that compared to group 4.
Absolutely, absolutely. And you're
Yes.
absolutely right. Like when you look at the time for when they drop below the 20%, it's yeah, very interesting. Very interesting.
Still have to analyze all this in depth.
Can you go to the survival
Sure.
next slide? Yeah, for me it's really interesting. Like the group 4, following on Peter's comment, is like if you look at the green curve, it seems like a majority of mice have stable disease or something between day 90 and then that and day 120, like 25 days they are fine, and all they drop after that. For me, I don't know how to explain that, because that's really striking. It's a good thing. There is something that's protecting the mice for 25 days. I don't know if you have any comment about that.
No, I don't. Maybe we can design some kind of study to look into that question in the future, in the near future. But I don't have any idea right now.
Have you ever done preemptive dose? So like you know, you get the tumors going and then you start dosing with compound. Have you ever tried preemptive treatment where you start dosing before and then
before the tumor inoculation?
Yeah.
No, I've never tried that. No.
I'm wondering if I mean, some tumors get very aggressive, and so when they take off, I mean, it's just too late. And I'm wondering, I mean, this tumor is considered an aggressive tumor, I guess. I'm wondering if you do a more protective strategy in these preclinical models, whether
you mean starting earlier here, treatment,
whether you can actually prevent tumor take. Yeah, it's just a thought. I don't know if you've ever tried that before and whether or not that's doable.
We've never tried that before, no.
Sorry. Looking at this curve, so one other thing is, you know, what you can see is that, you know, and I know the limitations to looking at you know, medians, right? But in terms of the 4-week treatment periods, you know, the median survival occurs, you know, roughly a month, maybe 5 weeks or so after treatment is stopped. And likewise in the group 4, the median survival occurs, you know, once again, about 5 weeks after treatment is stopped. So maybe it's, you know, treatment duration that is critical.
Yes. Yes. Also, I have to say this batch of cells or this specific experiment, they have a pretty high median survival. We usually have closer to 70 days than to 100 days. So maybe we wanted to have these curves like sharper here, you know, the black ones especially. This is like very little by little going down. Maybe in another experiment we can have better results, but I hope so.
So you think the cells are drifting in terms of?
Drifting? In
terms of their life, like typically you say it's about 70 days, but now it looks like they're surviving a lot.
Yeah, it looks like, yeah, I would like to have this sharper, steeper, you know.
Angel, can I ask another question?
Yes, Diana.
Diana here. So this experiment was done on nude animals?
Yes.
Yes, and the previous one, the 2009?
Not skid. Because
do you think that's related in terms of this, the curve?
I wasn't expecting this because in the nude, I was getting exactly the same curves compared to the non-skid in the last experiments. We've done a lot recently. And I don't know why this experiment is like a little different.
Okay.
Anyway, we have some kind of result, but Still,
So you
I'm not totally satisfied. Yeah.
So you think if the control had behaved like you typically see them, like at 70 days, then the outcome might have looked more interesting.
Yeah, 80, 70, 80 days is more frequent. Yeah.
Mhm.
So Angelo, you don't have a group, and did you ever try a group where you didn't have any vehicle or any treatment and see how long they survive? Do you know that?
Usually we don't give them. We only do this in this experiment because this experiment, with the previous experience with the 2009,
Mm-hmm.
we knew vehicle is important, and also these are oral, so we want to have the mice with the same oral. But we've done many experiments with no vehicle. The experiments, I mean, if there is no effect of the drug, the curves are like overlapping. The four curves, they would overlap.
Yeah.
We have many experiments we've done with many drugs, and the curves just you don't see the difference of one single day in the survival. So message is, I don't think the vehicle is important, and this is even less important because this is a what was it, like water, PBS?
Methocel. This one was methocel water, basically.
Yeah, methylcellulose and ultra. So there is no
methyl cellulose. Yeah, very mild. We've
Yes.
done that hundreds of times.
Okay.
Mhm.
No,
Oh, one
only
observation I have, sorry, is that the drug is not totally soluble. There is some kind of turbidity in the.
Yeah,
that's
at the high, but at the high dose, yeah, the high dose sometimes if you leave it, then you see a little bit of turbidity, but it's still okay to dose.
Okay.
Can I just ask one question? You were saying that the control group, the average median survival was longer. Normally you see something in the 70 to 80 range. But if I'm looking at this curve, that's just one mouse difference, is it not? Aren't we looking at like one extra mouse survived out to 92 days? If that mouse had died the same, I mean, it looks like to me like we're only looking at one mouse difference. So I'm not so sure about that. My takeaway from this is we're seeing a time dependency here. Obviously, we have a lot of talk stuff, but I think this is very interesting and very encouraging. Obviously, I'm biased to look for encouraging signs, but this is there seems to be some linkage or good signs of linkage to the time the mice are under the dosing. So probably something worth exploring further.
Mm-hmm.
Yeah, I agree. Time is important. When do you start treatment? When do you stop treatments? And how far from the treatment end goes this group? Because if there is no much tumor load when you finish, there is still a long time till the animal will die, right?
Yeah.
So we want to have like this balance with
right.
with a nice load in the brain, with a tumor, and still treating the animal, right? We want to have that.
I would actually curious, Peter, your thoughts on this. I would think that we don't want to start earlier because de facto then we're really getting further away from actually the clinical utility of the drug, which this drug is not going to be used on humans until, I mean, scientifically it'd be very interesting, but clinically the patients will not be dosed until they have a fair tumor load. So to the extent we want to have a linkage between the scientific preclinical studies and the clinical usage, we can't go really early.
Especially looking at this result, I agree with you. It could be interesting starting treatments closer to the day 50 and keep it for the 8 weeks.
I agree.
Yeah. And I think you make a good point. A lot of times in animal experiments, of course, when we've all seen this, right? We'll sort of cheat a little bit by starting, the day after implantation or something like that, which, what's really going on there, right? So yeah, I think a potential criticism for any result occurring, seen with earlier treatment is that one is sort of biasing the results.
I have a question. This is Al. I mean,
hey, Alice.
It's very cool, and thanks so much for helping on this. It does seem though that if we have to take a very high level, adding the molecule extends the life in some way, whether we dose it early or late. So that's a very cool result, and it's with this one cell line, of course, in this one system. My question to the more experienced folks on hives: at what point do you say, okay, the mouse has taught us what we need to know? Do mouse experiments ever inform in people about dosing regimens and when? Or have we can we declare mission accomplished thanks to Angel and say, you know what? Inhibiting this signaling pathway absolutely has an effect. It doesn't melt the tumor, but it absolutely extends life to various extents. And then we say, we've done it? Or are there, and I'm really speaking to Peter, who knows much more experience, do we learn more from doing more studies in the big picture? And this is asking out of ignorance.
Well, you know, if I just look at this experiment, I do agree with all the points made before. I think it's very intriguing results, right? But as we all know, with any experiment, you know, there are always you know, things that fall into the mix. One of them, you know, here is that the control group, you know, lasted longer. And I think Owen's point is also well taken, and maybe that's just one animal, but I do think that because of the toxicity that was seen, I mean, you know, 20% weight loss in animals is a common measure, right, that's used for that. At least for me, I would be very interested in seeing how a 15 milligram per kilo regimen for, you know, the longer period of time, 8 weeks, compares to control. And, you know, if you end up seeing a curve that's sort of somewhat like the green, whereas the blue performs more as these models typically perform, you know, then I think that's a extremely exciting result, and also replicates, you know, some of the interpretations that can come out of this experiment.
And just asking again out of ignorance, that kind of experiment where you have you modulate the dosing and the length in an animal, is that used actually to guide what a clinical trial would look like?
Yes, and with all the caveats that you know, it doesn't always translate one-to-one, and you know, you can find just as many examples where it does, and sometimes where it doesn't. But certainly the issue of chronicity of treatment is something that can come out of these studies. You know, we all know these animal models, right, are sort of engineered to get a result quickly or quicker. And so as such, right, they're really all set up for, you know, a fast-growing tumor and a quick effect to be observed. And so when that's not the case, I think that does inform, because, you know, if the goal here is to, you know, treat the patient chronically, you know, certainly that kind of regimen, even in an animal, I think is informative versus, you know, some short-term, you know, 5-day, 10-day, you know, treatment.
Okay, cool. Thank you.
This is Nicole Charles River. So just to add to that, I think we've talked about this before, but what's useful to go alongside this data often is some measure of target engagement. So if we have a biomarker, we've talked about phosphosmad being used in this project, but a biomarker where we can say we know that ALK2 was fully inhibited and that gave us an efficacious effect in this model, then that's really valuable. And we can use that dose at which you engage the target to try and predict doses to the clinic for humans from the target engagement data. But we know that it was a dose that was efficacious in an animal model. So that making that linkage is really important. And I guess it's something we haven't really done yet, is to try and measure the phosphosmad in an in vivo setting at a given dose of the drug, so that we can make that link.
Yeah, okay. That's good. So interesting. So what should be, I mean, next steps? I mean, should we just let this go to completion, or should we start planning for like a 50 mg dose longer, or should we just leave those discussions for another time?
I would suggest we discuss by email about this. My suggestion is to repeat the study, doing the longer treatment, also with the 50.
Yeah, that sounds like very good advice. Angle and your team, thank you very much. This has been incredible. I don't think everyone on the line appreciates the fact that you guys were actually in lockdown and you
Yeah,
started this experiment
I wanna
just before.
appreciate Leire. Leire was here dosing the animals during the lockdown every day, coming here, and everybody was hidden in the houses. She was coming here, so I really want to say thank you to her.
And we really want to repeat that too, because that was fantastic. The results of this experiment are very intriguing, and we really appreciate it. And I really hope that next time I will not book a vacation to Barcelona in the middle of a pandemic, and that I'll actually be able to make it next time.
Larry, I commit to send you some Canadian maple syrup. You'll get it within a month.
Oh, thank you.
All right.
For Leire, thank you.
Okay, thank you, Ango, for that overview.
Bye, thanks.
And good, yeah, please send us the slides when you get a chance.
Yes, I will.
Thank you very much. Okay, if any more questions for Angel, let's turn it over to Diana to get an update. So,
Can you see my screen?
yeah, it's all good. Looks good.
Okay, great. So, hey everyone. I think this has been the lockdown experiment phase for everyone, because just like Angel, I injected the week on the Monday, and on the Friday they told us that we needed to shut down. So it's been an interesting couple of weeks. I think it was a year ago, I tested both 2009 and 2163 in our patient-derived cells, including the HSJD, the DIPG7 that Angel just mentioned. And what we saw at the time is that the 2163 compound was definitely much potent than the 2009. And we also saw a differential in terms of sensitivity between the mutant and the wild type lines, especially with the 2009 compound. So these were some interesting results, and we thought it was a good idea to test this in vivo. So together with Angel, we planned the experiment that he has done in HSJD DIPG7, and I've used a different model. So last year, this paper from Oren Becker came out, and they used this RCAS-based genetically engineered mouse for the ACVR1 different mutations, the 206H, but also the G328V in combination with different genes. So the specific cells that I received have the RCAS-CRE, PDGFA, 3.1K27M, and the ACVR1R206H. So when Oren does this model, he puts this in black sixes with a nesting TVA P53 flux background, and they get tumors between in a couple of weeks. One of the things they do is then they take the cells from this tumor, they put it in culture, and they establish in vitro, and then re-inject in the model. So I received these cells, and this is exactly what I've done. So I've injected these cells in NSG animals. When I did this experiment just to see what was the survival about, I think it was September last year when I first injected them, we injected them in NSG animals, and we have Jessica Bolt in Simon Robinson's team here at the ICR that we collaborate with, and she does the imaging for us by MRI. And as you can see here, the tumors are visible by MRI in the axial and the coronal plane. There's a huge tumor, very cystic, and also it's taking part, going up to the front and also to the back where the pons are, just next to the cerebellum. And this is a staining. Sorry, it's not the best staining. We optimize this further now, but it's what I have to match the MRI. That we used the K27M antibody. And you can see here, it does stain for the tumor, because differently from the patient-derived models, we can't use the anti-human nuclei, which is really, really nice staining, but it doesn't work here because it's mouse on mouse. The other thing with this model is that the ACVR1 is tagged with an HA, and the histone H3B is tagged with a flag. So we can actually stain for these two with these two antibodies and see the tumor burden instead of using what we currently normally use, which is the anti-tumor nuclei. So the experimental setup is very similar to what Angel just described. So I injected 56 animals just beginning of March. We started on day 21. We just MRI'd all the animals, and I've collected the brains to assess tumor burden. And then we had 4 different groups. Group 1 was the vehicle. Group 2 was 100 mg per kilo per day, treated for 4 weeks. Group 3 was same dose, but for 4 weeks. And then group 4 was 50 mg per kilo during 4 weeks. At the end of the 4 weeks, we collected 4 brain and plasma for 4 animals per group, and also the animals were scanned by MRI, and the idea was to do the same at the end of the 8 weeks. So this is the weight loss per group, and these are the different from startup treatment, so week 1, 2, 3, and up until 6. And what you see here is that the vehicles are starting to go, even like at week 3 up to a few weeks after treatment has stopped. The 50 mg per kilo are quite happy throughout treatment and after. And the 100 mg per kilo, again, we have animals losing weight, but especially the one that continued after 6 weeks, you can see here, you know, they at week 5 and 6, they started to drop. And what I also noticed, it was not only like Angel just described the weight loss, but the animals really looked unwell. So I thought not to lose more animals and not to lose the whole experiment, I stopped treatment at week 6 when it was supposed to go for 8 weeks. And I just wanted to discuss a little bit here just to see what you guys think. And I think Angel also touched on that when we did the tolerability, we used the old formulation, which I'm calling DMSO and PEG and Twin 80. And Angel also saw a bit of toxicity in the HS when he did the efficacy study with this compound. So they had to stop for a week and then continue after. And at the time, I think we said that, oh, it's probably due to the vehicle, but maybe there was here a combination of the higher dose and also the vehicle that was being used. The other thing is with the new formulation, which is definitely much better than to dose the animals by oral gavage than the previous one, and much easier to make the drug, and it solubilizes perfectly. As I said to you, I had to start treatment at week 6. And one of the other things that kind of gives me, I think, the idea that it might be due to some toxicity in the highest dose was that the sick animals by MRI didn't really have a huge tumor, or which again is not is not to say that there wasn't a tumor, but it was definitely not a huge tumor that would give symptoms to the point that the animals had to be culled. And also the few animals that survived after I stopped treatment, they kind of recovered. So in 1 to 2 weeks. And again, this is just me observing the animals. I don't have, you know, actual data to show you, but just some observations that I thought I should share related to the drug. So as I've said to you, all the animals were scanned by MRI. So on day 21, just imaged them all. And what you see here pointing out is just the site of injection. This MRI was done because at this point we can't really see tumor. So she scanned them so that we could be sure that there wasn't like one animal with a big tumor and an animal with a very small tumor, and we could say that all the animals seemed quite similar, so we can randomize them and we don't have like outliers. And then she scanned them on day 46, so 4 weeks after treatment. And what you see here is the start of something there. This is an animal that has been treated with the vehicle. And then at 8 weeks, and you see this huge tumor coming up in the pons area, just below the cerebellum. So the idea here was to actually assess tumor volume comparing treated and untreated animals. Because at 4 weeks, it seems like we can't see much tumor. I think we'll have a couple of examples like these ones to show, but I'm not sure it will be the best measure of tumor burden for the study. But again, this data needs to be all put together, and really every single image needs to be measured, and then we'll have the complete data set, and we'll be able to say if it's possible or not. But this is just an example of one of the animals. And then the survival, which I think is something that was definitely unexpected. Not that I wasn't expecting something to work, but this is a thing. I've never seen this before. So what we see here is that all the animals, most of the animals of the 100 mg per kilo group are going down together with the vehicle. And then a couple of them survive a bit longer. So these are the ones that I say that then after I stopped treatment, they looked better, they kind of recovered. And the 50 mg per kilo are still going. And we have an increase in survival of nearly double, of 50 something days. Of course, we still need to get all the data and be sure that this is why this is happening. And there's a couple of things that still need to be done. So as I've said to you, at the end, at day 21, before starting treatment and after the 4 weeks, 2 hours post-dosing, I collected brain and plasma to assess tumor burden. And we are currently doing all the histology. The H&E is mostly complete, and we're staining for the K27M antibody, the HA, and the flag. That stands for the 3.1 and the ACVR1, and we'll be able to assess tumor burden using a software called the Definition, so it's just counting all the cells that are labeled. And then, as Angel, the digital PCR doing for the ACVR1 R206H mutation. And that's all still ongoing, so I don't yet have the results for that. And then we also collected samples for PK and PD, plasma and brain, 4 animals per group. The PK is going to be done in Toronto, so I need to ship all the samples to you guys. And I'll run some Western blots for Phosphosmat and ID1 for all the different animals that have been treated with drug and the vehicle for comparison. And I just want, this is the team, and Valerie has been doing all the histology, and Jess is not here, but she's done the imaging of all the MRI. So I just wanted to say thank you to them. And that's it.
Thank you. Really some exciting data, I think. Okay, so let's open it up to the floor. I mean, anybody have any burning questions for Diana? Thank you, Diana, for that overview.
Deanna, just a quick question. First off, thank you very much. I mean, again, I really hope we don't have to send people to risk their lives for our animal studies in the future. But I greatly appreciate everyone going in and the effort here. This is, the data here is really an eye-opener, and in line with what Angle said here today. We might be seeing some interesting signs about the series and some things to understand about the tox and the dosing, etc. But it's fascinating. Just a quick question with regards to the study you did previously, and we did not see a big extension of life. At that one, we were doing 100 mg per kg in the old formulation, but otherwise it was in many ways the same experiment. It's really looking like the original experiment was overwhelmed by either the tox of the possible tox of the compound itself or the formulation. Is that a fair view of those two experiments?
So, well, Angel can tell you probably better, but from what I know from the HSJD efficacy study is that there was definitely some tox associated with it. We also don't, because when we do the tolerability studies, we treat the animals for 2 weeks and then we stop. And the animals are fine. Here we're going further, right? We're doing 4 and 8 weeks. So if this drug is getting into the brain or is accumulating, with all the dosing, which we've seen again with LDN 193189, where we could also treat for more than 6 weeks, maybe there are some long-term effects of the highest dose. But it's hard to say it's actually due to that, because these are only it is me observing the animals and saying, I think they don't look right. There's something not quite right here. But the observations make me believe, because they recovered and because the tumor burden by MRI is not huge, then there might be some toxicity. But in regards to comparing it with Angel's experiment, I would have to ask him, because I don't exactly know if he saw the toxicity in both the vehicle and the treated at the same time.
Yeah, I think for the last experiment, the actual treatment groups were looking better than the vehicle groups. That's why you got so concerned about the vehicle.
Medvin, do you remember? We repeated this twice. Once.
Yeah, when you had done the 2009 experiment, one of the biggest concern we had was the vehicle body weight loss. They were doing worse than the compound treated animals. So that's why we got overly concerned, the vehicle.
Yeah.
So you repeated the experiment then with the new formulation?
I really don't remember. No, we didn't. I don't remember if we
so
repeated
we didn't do 2009.
The 2009 was only once. Repeated the experiment. Okay.
Yes.
Oh, I didn't know that.
But we do 2163 instead. We had a choice between the two compounds, and we decided to go for the newer compound, which look a little bit more potent.
Okay.
So yeah.
Yeah, okay.
Any other question? Owen, did that answer your question?
I'm going to have to take a look at some of the historical data, but thanks very much.
Sorry, guys, I have to leave. I have to say goodbye. Thank you very much to everyone.
Thank you very much,
Thank you, Angel.
Thank you.
Metin, I had a question on this experiment.
Okay.
Danny, I think you mentioned that that there's still a lot of data analysis to be done, and you also said that your observation was for the sick animals, their tumor burden, either was lower or much less. Is it your sense, and will you be able to tell from the MRI scans, or at least a quantitative analysis of the MRI scans, whether tumors actually got smaller over time, even if for a certain period before they grew, or is it more that the tumor growth in the treated animals who survived longer simply they simply grew more slowly?
So by the MRI, as I said to you, because the tumor was not so visible at 4 weeks, it will be hard to determine. But I'm hoping by the other, by the histology and the digital PCR, we'll be able to say at least that there is less tumor in the treated animals than there is in the vehicle.
Okay, thanks.
And would it be true to say, because the tumor by its very nature is diffuse, imaging becomes, unless you actually have a nice localized mass, it tends to kind of just diffuse throughout the pond. And so that density that you might be looking for in an MRI, the tumor may still be there, but you may not necessarily get that localized density that you're looking for.
True, and with models like the DIPG-7, like you won't see anything apart from a shift in the midline until nearly at the end point. With this tumor, we do still see, even if it's a GEM, an RCAS model that should be, very homogeneous, we still see some masses, and it's much more dense than some of the CDXs and PDXs. And we do see these cystic regions. And also the other important thing is, because we scan so early, any alteration in the brain throughout is an indication that there is tumor present there. So if you see a small change in the brain and you have something to compare it from day 0, you can say that there is something growing there. Of course, then this is what I'm saying, at day, at 4 weeks post-treatment, it will be hard to say, oh, tumor volume was 10 versus 5, it will be hard to say that, but we might have some examples that we can show of animals that have been scanned throughout all the time, that, for example, the long-term survivals, we have scans from day 0 up to now, that we might be able to see something there.
Okay. It's good. So so next step. So yeah, you're going to ship some of the samples back to us
Yes.
for PK studies,
Yeah.
and it's kind of
Yeah.
nice. Speaking to what Nicole was saying, if you can get some, you know, phosphosmad and, you know, ID1 as markers and stuff to get a good sense pharmacodynamically what's going on, that would be good.
So
Are you also planning? Are you? Sorry.
oh, I didn't realize you're done. I was just going to ask Diana and Chris what they thought the next studies or follow-ups to this would be of most interest.
I think for it just depends on also what you're interested in seeing, and what if anyone else has tested the other, you know, 2163. But I would be interested in seeing what, if that's the final one of your last final compounds, it would be great to see how that works in this mouse model, because it's like a good comparison with the HSJD, the IPG7, because it's a very different model, right? But maybe
Yeah.
Chris can add to that.
No, hi, it's
Good.
Chris.
So we move on to straight again.
Go ahead, Chris.
Sorry,
Go
no,
ahead.
I was
What were you saying?
sorry, I was just going to agree with Deanna and say it would make sense just to tidy it up to have the 2163 done on this model as well. So maybe we could touch base about that and just think about
Yes.
precisely what dosing we did.
I agree.
Got that data on this model as well.
Good. Any other questions for?
Yeah, I would think, just for my 2 cents, I would be interested to see potentially a randomized between the two drugs, and to help us see if we can get an indication about which of the two looks more favorable, or if there isn't enough difference that we can see in an in vivo model to decide at this time which of the two molecules to move forward, or to accelerate. But this has been fantastic. I mean, it's those survival curves are quite something. These two, to me, these two in vivo studies have turned on our heads with some of the thought process, at least I had, that we had a very well tolerated compound that we could dose at high doses for extended periods of time. But now it actually looks that we have some potential signs of some toxicity events taking place, and that they actually show efficacy at the lower doses, which is quite encouraging, actually. So thanks very much. This has been quite exciting, and it's, as always, these experiments add 2 questions for everyone to answer.
I have a quick question for Deanna. Sorry, Metz. Yeah. Sorry, Metz. I have a question for Diana. So I was asking the PD marker, the phosphoSMADs. I know you are going to look at them at the end of the study. Do you have any experience to look at them earlier? Like what I'm trying to see, you know, like you can go through the whole experiment, but can we get the feel for the target engagement earlier than end of the study?
So for this specific experiment, I've collected just after treatment, so it's not end of survival, so after the 4 weeks.
Okay.
We have previously, not with this compound, but we have previously done time points in healthy animals, which is not great because they don't have high levels of phosphoSMAD 159, so any modulation is hard to see. So you have to do it in animals that carry the tumor. This specific model does have high levels of phosphoSMAD 159 that I've tested in the cells in vitro. So I'm hoping that at 2-hour time point that I've collected post-dosing, I will see some modulation of the pathway. And in terms of doing more time points in a next experiment, maybe it's something we can look at. Yeah, something to discuss. But in previous experiments, we've only done it at the end of the 4 weeks. and on day 0, so before
Yeah.
starting treatment.
Okay, good. Thank you.
Okay. All right. If no other questions, thanks, Diana, again for the update, and your team for the efforts that they have put into really get some of these data to us through this difficult time. So thanks again to you and your team for getting this done. Next on the agenda is Sukram giving us an update on some of the chemistry from Charles River. So you want to drive or you want me to load your slides? Which one?
Do you want to drive? I haven't got them open at the moment. So.
Okay, so I'll open up your slides. Oops. So I'll go back to share screen and go to show all windows. Can you see that?
Yeah, that's fine. That's fine.
Can anybody see that? Yeah. Yeah.
Okay. So just the first slide is just really a general update on what we've been doing. So over the last couple of months, we have had resource available at Charles River to make new compounds. So that's been really great. So we've got a number of compounds that are actually being shipped off today for screening. So there will be samples sent both to RBC, to the OICR, and also to SGC. So basically, once we had some discussions at the recent SAB, so following on from that, we sort of were aware that there was concern by the SAB that perhaps potency in our lead molecules might not be quite as good as we would like. So we had some discussions with Methvin and Owen more recently, and we agreed that we should redirect the focus towards improvement of potency in both the main and backup series. So as I say, until recently, we'd been focusing more on trying to improve the brain penetration of the compounds that we were working on within the Blueprint series. And we've made some progress, and we were still focusing on that. So most of the new compounds that will be sent for screening will be from that set. So more recently, we've been starting to look at what within that series. We've got some compounds that are very highly potent, but they didn't have very good brain penetration. So trying to see whether we can get a better balance of improved potency, but still trying to improve that brain penetration. And I've done a very preliminary data mining of the activity in the main series to see if there were any other opportunities either within that series or areas that we might be able to apply into the Blueprint series, so just to see how to further improve potency. So obviously we have a slightly more limited screening cascade now. So we're the only group that are doing any synthesis. We will get the ALK 245 potency from RBC. Hopefully the SGC guys at SGC will be able to come back online and do look at the cell potency at least in the nanobret assay. And we agreed that with Owen and Methvin that we might be able to get some CACO2 data on a few compounds that might be looking interesting. My other concern that occurred to me more recently is we probably also need to look at metabolic stability of selected compounds only, but I'm not quite sure how likely we are going to be able to get that under the current situation, as there's not a lot of resource available. Obviously, or most of the resource is obviously focused on the models and the progression of the leading compounds. So next slide, please. So the next couple of slides are ones you've seen previously, which are just what we were doing to look at the follow-up to the Blueprint series. And I leave these in as just as a reminder from because it's been a little while since we've been talking about this. So M4K 3007 was one of the lead compounds from the blueprint series. Very potent for nanomole. It's also very potent in the nanobret assay and is quite selective. The unfortunate thing about it was that when we looked at the PK, we had quite poor brain penetration, and that was consistent with what we'd seen from the permeability assay. So we saw poor permeability, quite moderate permeability, but there was a reasonably high efflux. So we sort of looking at this, we decided that the molecular weight and the pKa are likely to be the main contributors to the poor CNS penetration. As far as pKa is concerned, you've got the two basic centers which are highlighted, and the initial plans were aimed at overall improvement in the phys-chem properties, while obviously ensuring that we don't lose too much of the potency and selectivity. There's a progression that we've gone through, and the leading compound now is on the next slide. So the leading compound now really is M4K 3093, but as you can see, we've lost a little bit of the, even in the enzyme potency, and I haven't put on this slide, but the cell potency in the nanobret assay has gone down considerably more. However, despite that, we are seeing a good, an improvement in the PK profile compared to 3007. We got increased exposure. We saw a lower clearance than we'd seen for M4K 3007. And we also saw now the brain to plasma ratio had gone from 0.16 to 0.48. So we are seeing some improvement there. It's probably not quite where we want to be. So we've been looking more recently at doing some further modifications around that structure. If you look at the next slide, please. So looking at the two main areas, we've got the cyclopropyl amide of the piperazine at the top, which has been pretty consistent throughout most of the compounds. We do know that from some of the modeling studies and some of the work that's been done at the SGC that the cyclopropyl group in one of the mutations is involved in a clash, and therefore that it's probably not ideal. So we've started to look at is that amide required, and one of the compounds we've made is obviously taking the amide out, replacing the piperazine with a biomorpholine. And see whether that retains activity. Obviously, we've reduced the pKa there. Then the majority of the other modifications that we've been working on are shown on the right-hand side, where there's sort of modifications to the tail. And all of these are pretty much result in a reduction in the pKa. As you can see from the indications there, we've made substitutions on the piperazine. We've looked at some of the fused systems, and we've also looked at some of these pyrazole type compounds. And part of this was as a result of looking at some of the more active compounds in the main series, and I'll come on to that a little bit later. But we've made a range of compounds, and basically we've been trying to modulate the phys-chem properties, and the majority of these are actually ready to ship off to RBC and the other places. The next slide, please. Okay, so I was looking at what we might be able to do to improve the potency. So we've got 4 compounds that come under the blueprint. The top 2 compounds there, 3007 and 3078. They're both within the blueprint patent, but they are very potent. They're very active in the nanobret assay. As I say, when we made 1393, we saw a significant reduction in the activity in the nanobret assay. The compound underneath 3120 This is from the second blueprint pattern that we were looking at. This is a smaller molecule. What we've seen here is, although the enzyme potency is in the same region, we've got very potent molecule. So if you can click on the next. So if you look at the top areas, so in the top bit, we've got the main difference between things like 3078, 3079. We've got, 3007 and 3079, we've gone from the ethoxy piperidine to the piperazine. And although, again, the enzyme potency is not significantly reduced, we get a reduction in the cell potency. And in the next highlight, 3120 to 3110, although we do have a slight difference in the carbamate group at the top, the nitrile seems to be the main modification. And it's interesting to note that some of the more active compounds in the main series also have a nitrile on the central pyridine. So what we're not quite sure is what, at the moment, this is sort of quite early stage and not quite sure what the actual cause of these. The unfortunate thing is that, well, certainly the top two, they have much poorer PK profiles, but they have much better potency. So as always with MedChem, it's a balance of getting the right properties. We don't have a PK on 3120. I think that was stopped at lockdown. So we don't know whether that compound is any better or worse than some of the others. But it obviously, compared to the main blueprint series, this has now got a bidentate hinge interaction rather than monodentate interaction. So there are some other differences which could be enhancing the potency, but you can see from the compound on the right that, you know, you do lose the cell potency without that nitrile. So it's just stimulated a few thoughts of some things that we might be able to do around this area on the next slide.
Sue, it's my apologies. Can you educate me a little bit here? 31
possibly.
It shouldn't be hard, trust me. 3120 looks, obviously it's fairly interesting. The IC50 is 13 nanomolar.
Yeah.
The nanobret is actually showing
yes,
quite potency. Why? What's the thought here about those differences in?
I would say that's quite unusual that you would see it more active in the nanobret, I say, and it's probably something we would want to repeat. But obviously, SGC have been in lockdowns, or they've been totally involved in COVID related activities since we got this result. So you would normally expect a small drop off between enzyme activity and cell potency.
Yeah, that was what was going through my mind. And
I mean,
okay, very interesting.
Bear in mind too, the enzyme assay which we're running at RBC, anything between 1 and 10 to 15 nano. I'm starting to believe that we're at the bottom of that assay. So we might need, if we might need to see more improvement, maybe that compound is a lot more potent than we think it is. We might need to start looking at some sort of binding, some sort of discovery type binding assay.
I'm not sure
to figure that out.
The limit in the potency, because we've seen some compounds from the main series where we've got enzyme potency less than, I think 2 nanomolar is their lowest. So they do say, because there's been some compounds that have been less than 2 nanomolar. So obviously we don't know what the true activity of these is. But yeah, I mean, you usually reckon 3-fold anyway. So so that first compound, it could be 4 nanomolar in the enzyme and 6 nanomolar, say, in the nanobret. But the important thing is the difference in the activity in the cell assay between 3120 and 3110. You can see there's quite a big difference there.
The other one of the important thing I was just responding to Owen is the cell permeability, right? So if usually you have a drop off because your molecules are not as permeable or effluxing out in the nanobret assay, but this may be more cell permeable. this compound.
Mhm.
I don't know.
Yeah,
Do we-
So
yeah.
which
That-
could be very interesting, obviously. From that's what jumped to my mind too.
I'm going to put a slight caveat in that.
Damn.
Compound
Damn,
3078 is less permeable than some of the other compounds in the CO2 assay.
Mhm.
So
Yeah.
I don't know that these compounds, some of these compounds have only moderate permeability.
Mhm.
So the 7 is not that, it's not really highly permeable.
And 3120 is the first one I'm going to call it in this sub-series. Is it not? Where there's you've truncated.
We made 3 compounds from that patent. And the 2 of them are shown. The other one had a pyridol. It was like 3110, but it had a pyridol on the right-hand side rather than a pyrazole. And that was even less active than this one. I can't remember off the top of my head the exact activity, but so that we only made 3 compounds. I'd really like to see the permeability on that, because I've had experience of that type of structure before where they're not particularly permeable, but it must be. Must be permeable.
Mhm.
Unless there's something in some of the compounds that we're not seeing from something like the CACO2, which is effluxing them out of the cells, which you might not see in CACO2, I think.
All right. Well, looks like we have a lot of questions and not a lot of available resources right now.
Yeah.
But that's this is one not to forget, I think.
Yeah, absolutely. So on the next slide. So these are just a few of the ideas. So if we stick perhaps with the first blueprint series type compound, can we mimic the effect of changing the piperazine, the ethoxypiperidine to the piperazine is to reduce the pKa. Can we keep with the piperazine but incorporate a group somewhere on the piperazine ring rather than, obviously you can't come off the nitrogen. So there's just a couple of thoughts I had there. And at the bottom, it's unfortunately synthetically very challenging to put something like a nitrile onto the core, but can we reach into that area from a group, or from the pyridol or the pyrazole type group off the side and enhance the potency there? So looking at the docking, those 2 groups don't seem to be generating any particular interactions. I think that it must be something about the phys-chem properties. But the trouble is the compounds that have the better cell potency seem to be the compounds that have the poorer PK profile. So again, it's down to that balance. So these are just some preliminary thoughts. On the next slide. So this is just really looking again back into the main series. So I did a bit of just looking at what were the most active compounds, looking at what had good ALK2/5 selectivity, and also including in there the nanobret EC50. So again, this is the question of what is making these compounds more active. So if you can just click one more. So the first one is that nitrile again. Is this doing something? That appears in a number of these compounds. Now, initially, my thoughts were that nitrile was resulting in a loss in selectivity in compounds like 2250 in the middle of the top row, where you've only got 9-fold selectivity. However, if you look on the bottom row, you've got 2214 or 2220, and both of those have got at least as good selectivity over ALK5 as 2009. I've put the date of 2009 up at the top, on the top. And you can see you've got better nanobret activity. And I'm not quite sure why some of these, there might be metabolic stability, there might be CACO2 data, which I haven't got at the moment, but this was just looking at it from an activity.
It wasn't.
Sorry?
Yep. So 2214 and 2250. I think 2214 was part of our short list of 10. That was looking really good. It was one of the ones we were really excited about. And then it's that moiety that started hitting CYP. It was hitting all the CYP enzyme really hard.
Okay.
So that's how that one dropped out. But that was one of our strong candidates, actually.
So yeah, I mean, I'm sure there are valid reasons, but I was thinking more in terms of from a perspective of what knowledge can we gain that we then be able to take on and make some newer compounds.
I agree. Yeah.
Click on again. So these groups don't appear that often in compounds. So these tail groups. So the two on the right hand side only appear less than a handful of times. The two on the left hand side, yeah, they do appear in a number of compounds, but it's not a huge number. So again, is there something we can learn from these different ones? Again, either taking those types of groups back into the blueprint series, or is there some more compounds that we should make with those tail groups? And the final one, I think, was, click on, yeah, so it's the amide. Obviously, that appears to be beneficial in these compounds. And this is, again, where I think there's a disconnect between what we're sort of anticipating from the cell activity and the permeability and getting into the brain. So these compounds, obviously, we know that these amides have poor brain exposure. And I know that's some of these have not progressed, and I know there were 2 of them in the final 5. But yeah, it's interesting to see if there is some features that we can use from this. I think there was a final. Was there? I think that might be my last slide. Yeah. So that's where the idea of this was to just start to look back at the data and see if there's anything new, because with a view to increasing potency further. What can we learn from the data that we've got, and what we do with it.
Methvin, did we come away thinking the nitrates were causing the CYP inhibition? CYP activity? Or was it the methyl? Do you recall? Or have we not got that far?
It's the cyano. So we had similar analogs of 009 with the nitrile. That was actually a lot more potent, more selective, and then they were potent across all the CYPs. That's where that nitrile became a problem. So we got very concerned. That's where that compound fell out. So we saw activity across all the CYPs, and we think that was one of the key criteria we wanted to see for the first batch of compounds coming through.
Yeah.
Since if you were thinking of the likelihood of using these compounds in combination, we wanted to mitigate any sort of CYP liability.
Could I ask a question?
But it's a good point. We could go back.
Yeah.
So,
Yeah, Methvin, I was just wondering, in view of that, we are considering going forward with monotherapy as one of our possible presentations. Isn't that correct? At least initially in the clinic.
That's the strategy for proof of principle, yeah.
So in view of that, is it worth just revisiting the idea that CYP is a major liability, given the severe situation with these patients?
Yep, I mean, if one of these compounds start looking very stellar in the anti-proliferative assays and in vivo, well, of course, we might start to rethink that. Like one of the SAB members said is, if we could start getting more potent compounds at the target, if this is one way to do it, it's something we are open to consider. Yeah.
And even short of that, just consider, as if we're looking at getting a more robust efficacy readout, maybe 2214 would be an ideal candidate to really test, what are the limits of ALK2 inhibition in these models.
Yeah, that's a compound we had an eye on for quite some time. Yeah, and that's why it's a good point to go back and revisit, because at that stage, our criteria, things that we're looking for, you know what I mean? We wanted to overcome hug, we wanted good potency, we wanted good selectivity. Now we have those candidates and we're evaluating them. Now it's a good time to go back and revisit what is it that we can tolerate in the profile of the lead compound, you know, at the expense of a property that we might need to see. Could be improvement in potency. So these are things to go back to. That's why we're mining some of the old data now, based on some of the comments that are coming up these days.
Yeah, I like Sue's suggestion to go back and revisit this, because maybe with some tweaking we can perhaps not get rid of the SIP completely, but you know, if we could drop it a few fold, we might be back in the sweet spot.
Yeah. And I know Peter's going to talk about the SAB feedback in a few minutes, but you know, when they took a look at our molecules, they zoomed in on potency, and low single digit nanomolar potency was what they flagged, and what they thought was a weakness for some of our other compounds that we had in our cold group. I'm not sure we spent a lot of time thinking about SIP. We definitely they definitely were not as concerned about the HERG issues as we had. That's not a fair way to say, but they minimized, in their view, our view, the risk of the HERG, and you know, really wanted us to think about selectivity and nanomolar of potency, which 214 and 218, and I guess 220 also are really interesting candidates for. Anyways, this is it's you know, Sue and your team, thanks for revisiting this and bringing it to us again, because it's sometimes you forget, and it's good to come back and think about these things especially after the feedback we got from the SAB.
Yeah, and that was the reason for doing this, really. It's just because of what they said.
Yeah, no, great, it really is. And it's I think we need to this is probably not the right time to do it, but I think we need to start thinking about what to explore here again. David, Methvin, Sue, your team, etc. Peter, obviously, let's think about some of these molecules.
Yeah, it may be preempting the discussion at the end, but I would enjoy hearing Peter's comments on the SIP liability in the context of this indication.
Yeah.
Okay. If there are other questions for Sue, I mean, I had one last quick question. So, you're planning, you see, for 3120, you said you wanted some PK done for that. So maybe if you have enough of that sample, you know, you could probably send some of it to us, and you know, when a slot opens up for rapid PK. We have it already?
Have it. Yeah.
Okay.
I think you were about to do the PK when lockdown happened.
Okay. Yes.
So
we'll check.
So
I'm pretty sure that we shipped it. We would have shipped it to you when we made it.
Yeah. Okay. Yeah, so we can get a sneak peek at the rapid PK at some point when a slot opens up.
That would
we can
be
run
helpful.
that compound. The other question I had is for the impact of the flow you had on the bicyclic wing. Were you thinking of adding that flow to 3120 at some point to see if it helps a lot of the fizz properties too?
We probably weren't, because basically what that fluorine does in the first series is reduces the pKa of the piperazine across there. That, because you've got that nitrile on there, again, you probably don't need that. The pKa of that is much lower. Type of hinge interaction, so you probably don't need the fluorine on there, but we can put other groups other than the nitrile on that second group. We just can't do that in the top series.
Okay. All right, that's good.
Okay.
Thank you for that overview. Some good ideas to go forward with. Okay,
this is Al. Can I make one comment, please? So Lyra and Deanna are going to get maple syrup. Sue, unfortunately, you're a company. I can't be sending gifts to company. But what I would like to point out to everybody, I mean, the Charles River commitment to this has been phenomenal because if anyone's worked in corporations, the initial excitement is almost not easy, but to get a commitment from a company to a new idea is not as hard. To get sustained commitment is incredible. And, we have to remind ourselves how cool it is that Sue and Nicole and their teams are working with this, because it's harder and harder as time goes on to maintain the kind of commitment that Charles River is giving. And so anyway, thanks a lot. And I just want to sure everyone on the call knows, but it's good to remember, right? It's so hard for companies to sustain, much harder than to commit the initial excitement. So thanks a ton, but you're not getting maple syrup.
Thanks, Aldo. Really appreciate that. Thank you.
Okay. All right. So let's move on. We have about half an hour left. We should be able to So we're going to go back to some of now the comments that we got from the SAB and the TPAC for updates. So if I could just load those up. So stop share. Oops. So Peter, you want to go first? So yeah, so for the benefit of the team, so in May, like as I said, Peter came on board with all of his expertise and background, and one of the first things he did, which was really good, was assemble an SAB to starting to look at the DIPG project and the lead compounds, and he did a nice job pulling a group of people together. And then we had an SAB meeting on the 22nd of May. And then 5 days later, we had a CTIP presentation, that is to the OICR board that was funding part of this project. So I let Peter go on and give you an update on some of the feedback that we got from the amazing SAB board that he assembled. And then I'll give you some of the feedback from the CTIP updates. So Peter?
Sure. Thank you, Vincent. So yeah, this was definitely one of the more unusual SABs I've been asked to put together. When I contacted the folks here, and I'll just describe them briefly, but when I contacted them, I said, well, because of the COVID situation, we're not asking you to do any travel, so you don't have to think about that. And because M4K is M4K, you won't get any honoraria. So really, this is nothing that you have to commit to. But there was a lot of enthusiasm in terms of the folks that I had connected with. And Greg Riemann, you see here, has a leadership position in the FDA. In addition to his title there, he's actually the head of all issues in pediatric oncology at the FDA, and former head of the Children's Cancer Group and former head of the Children's Oncology Group when CCG merged with the Pediatric Oncology Group. And besides the fact that one of his prior foci in the clinic was in brain tumors. Alan Olaf is at GlaxoSmithKline and has been involved primarily on drug discovery, less so on drug development, but on drug discovery for the vast majority of his career, which started off at Merck and then, gosh, over the last 20 years has been at GSK, doing not just oncology, but in other therapeutic areas as well. Uta Bartels is, I think, well known to many of the folks on the team, as she has been working with this program from her position at Sick Kids. And Uta recommended Cynthia Hawkins, who's a neuropathologist and translational person who works with Uta also at Sick Kids. Miriam Falati is, and the last two individuals here, Miriam Falati has had some prior association with this program, so knows it some. And she's at Cincinnati Children's and heading up the Brain Tumor Center there, although my understanding is she is moving to a new academic position this summer. And then Catherine Warren is at Dana-Farber Cancer Institute. She's the head of their pediatric neuro-oncology program, and previously she was at the NCI in the pediatric oncology branch there, heading up neuro-oncology. So Kathy has no prior background association with the program, so is seeing it for the first time. Okay, so go ahead, Metin. The next several slides relate to the questions that we posed to the SAB, and in blue is a short summary of their response. But before I dive into the details, I'd say overall, the reaction of the SAB, some of whom knew this program from before, but many others, again, did not, was very positive. And I think, especially for those who know the disease, DIPG, very well, understand the current limitations in treatment and the lack of therapeutic options other than radiotherapy for these patients. A lot of enthusiasm for the excellent work that has been done in getting us to the 5 inhibitors that were presented to them, as well as the desire to see something come out of this, and a lot of encouragement to continue moving forward. So that's sort of in a nutshell their response, and again, enthusiasm and encouragement for this project to continue forth in the direction that's been currently set. Okay, so I think one of the objectives in our meeting with this SAB is to say, hey, look, over the last couple of years or more, gotten 5 leads, and the team had looked at these and wanted to start coning down the leads as we move toward a candidate selection, which will still, of course, require additional data that has yet to be generated, but still moving down there. So essentially, what does the SAB think about the progress made, and do they agree with the team's evaluation and rationale for moving from 5 to 2, the 209 and 2163 as the most viable candidates to continue forward with. So we had them review the TPP, and all of that. I think this team knows well. There was general agreement that the criteria, the selection criteria, and the selection parameters that have been set are appropriate, and really no specific recommendations from the SAB to make any major modifications. We did focus next on the hERG characteristics of 2 of the compounds, and saying that they were these were particularly favorable for hERG, and we'll come back to this as well, but that they had a lower predicted CNS penetrance, and what did they think? And this will come up later with another question that we asked. But although, of course, from a clinical perspective, QT prolongation is something that is to be avoided, and looking at her characteristics remains part of the regulatory review. The overall perspective was, well, if this is yes, this is one parameter, but it really ought to be balanced with respect to other risks to the patient. And in this particular case, given the lethality of the disease and the lack of therapeutic options, I wouldn't say it takes necessarily a complete backseat and it should be ignored, but it really should be thought of in context of that. So again, shouldn't be ignored, but really, essentially, these patients have other things more pressing to worry about in terms of their immediate clinical course rather than the risk of QT prolongation. And so they agreed that CNS penetrance, obviously, in this disease is very critical. And so they agreed with the team's assessment with these two particular molecules. Any questions on these before we move on to the next slide? And I'll just say the first several questions actually all relate to the rationale behind the coning down of the molecules from 5 to 2 in various ways. Okay, so then if there are no questions here, let's move on to the next one. So continuing on with this theme. Sorry, I think.
Did I go too far? Yeah.
Yeah, back another one, please. Yeah, okay, there you go. So, all right, so with 2117, also the rationale for deprioritizing that was its poor anti-tumor activity, and basically, what do you think? And this goes back to a point that Owen made earlier. So in looking at all of the molecules, there was the general view that, of course, anti-tumor activity is critical, and the feeling that although certainly the two compounds that are recommended to continue forth were and are supported by the SAB, they also felt that there could be additional work done not only on a backup series that, of course, Sue had just described with the blueprint scaffold, but that even with this current series, that there can be additional medchem work to improve upon, further improve upon potency. Because and really driving it down to single-digit nanomolar should be a goal that we have, because certainly, as we see in this case, and certainly with other molecules and other models, as you move from a biochemical potency to cellular potency in whatever whole cell phenotype one's looking at, and then further into in vivo models, and then in the clinic, that there is a general loss, if you will, in terms of that potency. So driving it down as much as reasonably can be reasonably expected would be a good thing to do, and so that brought up the discussion points that Sue made earlier in terms of relooking at the current series to try to further improve on that as well. Protein binding on the next one, protein binding for 2163. It is a bit of an outlier, if you will, in terms of its protein binding characteristics with respect to the other 4 molecules. And did the SAB feel that that was a significant liability that should be part of the consideration? Basically, should we not continue forth with it? And certainly, as we all know, having a lower binding protein binding is a good thing, but by itself, the characteristics for 2163 did not preclude its progression. And I think part of the SAB's rationale there was looking at the in vitro activity that 2163 is already showing. Certainly, I think if that weren't the case, maybe it might have been a different view, but I think that sort of offset the measures that are in place for 2163 with respect to protein binding. And so really last, as part of this series of questions, you know, funneled down to, okay, well, you know, given all of this, do you agree that 209 and 2163 warrant continued progression toward a candidate selection? And indeed the answer was yes. And there was, I wouldn't say it was unanimous, but certainly there seemed to be a bit more support for 2163 than 209 at this point, based on seeing in some, but not all cases, right, improved anti-tumor activity from an in vitro setting from the standpoint of self-proliferation assays and induction of apoptosis. So that covers, and this, you know, to be fair, this is where we spent most of the discussion for the SAB. There are other questions that we'll get to, but again, before we move on, any other comments? Otherwise, I'll move on to question number 2.
I'm sorry. I had a question that just popped into my mind with regards to the protein binding. In the mouse, the protein binding, what's the similarity between protein binding in a mouse versus a human? Does anyone have any insight into that? We seem to be having interesting results in the mouse models. I'm just wondering if there would be any correlation or anything we can take from that mouse protein binding to human.
Yeah, it can be very different at times. I mean, I've been in programs where it actually got worse as you move towards human, but see a lot of refraction in mouse. So you do get differences sometimes.
Okay. Well,
right. I think, Nathan, you know, the point to be made is to understand what you're seeing in mouse models, one needs to know that, but ultimately one should be measuring protein binding to you know, human serum proteins.
Mm-hmm. Okay, thanks.
So this is back to, you know, QTC, and then this is sort of going to the flip side, where some of the molecules have what appears to be more of a QT liability with respect to hERG, and, you know, essentially we sort of talked about this before. Yeah, you know, understand it, know what it is, unless it's horrible. You know, try to pick the better compound with respect to, you know, anti-tumor activity and other pharmaceutical properties, again, but not necessarily ignoring it. Question number 3, you know, is one that really is it's a liability for this disease, right? In the sense that the preclinical models, both in vitro and in vivo, and certainly in vivo, are less well characterized, less known to be predictive of the clinical setting than in other, you know, disease or other tumor types. And so basically, you know, are you comfortable with what we have, and the direction that we're going? And certainly there was some discussion amongst the SAB in terms of you know, their understanding and experience with the limitations of these models, but they felt that it's still important to generate these data. In our slides, we presented to them the 3 lines of in vivo work that are being done, and you know, earlier of which we have seen some of the data. At that time, we had very little of that, so they just knew, well, these are our plans in terms of moving forward, and they were very supportive of that, and certainly interested in seeing what the results are as they come out. Now, you know, one thing that they did say is that because of the disease, you know, maybe there's a bit of a lower bar, if you will, in that, you know, even if the in vivo results are less than, you know, absolutely compelling, and, you know, in other tumor types, one would have a higher bar, right, based on current treatment options and understanding of the biology of the disease. But here, you know, they want to see what the data shows, but there may be a little bit of a lower bar in terms of that. And you know, rather than in vivo data being used as an absolute go no go decision, it really should be evaluated within the context of, you know, the entire data package and how the molecules behave, you know, in other settings as well. Okay. Any questions? All right. And then moving on, you know, again, this is sort of a follow-up of this in terms of, well, given the limitations, you know, are there other things that we haven't presented to you that in terms of pharmacologic profiling that you feel we ought to be doing? And there was some brief discussion, but ultimately, you know, nothing that really jumped out to say, oh, well, you're missing this and you know, you have to do this. The next question related to something I think Dave mentioned earlier, and you know, there's been discussion about before, which is that you know, with the understanding of the biology being what it is, it may be that even if there is a selection of patients, you know, who have mutations in ACVR1, the clinical outcomes for patients who are treated, you know, may not be the same as you know, some other targeted therapies and, you know, lung cancer, melanoma, et cetera, and that, you know, we are already thinking about combinations of an ALK2 inhibitor, you know, with other things, and in the way the question was posed, you know, we gave the example of a CNS penetrant EZH2 inhibitor or an EED inhibitor as a potential future combination, recognizing we don't have that today. You know, the SAB is the message, and this was alluded to earlier as well. The message was essentially, yeah, you know, a combination like that may be terrific, but there's so much that we don't know now, and there's still a lot to be gained and learned with a molecule that, again, if it meets all of the existing criteria to advance into the clinic, you know, even with what might be some lower expectations for what can be seen as monotherapy, that the SAB would, you know, would support moving ahead with the molecule in the clinic, again, as long as it meets all of the criteria that have been set forth that they were able to review. And certainly there was, I think, quite a fair bit of enthusiasm to look into whether there would be a molecule, whether resident today or to be identified in the future, that hits the EZH2 EED as a target, even if it's several years behind. Okay. And then finally, the last two, we asked a question about oral formulation. You know, not surprisingly, you know, a suspension given the population with attention to taste and taste masking if needed. But really, we didn't have a whole lot of time to get to this question. And then the last question with regard to funding, really, we didn't have a whole lot of time at all for that. And so, you know, what's there is sort of what's obvious. But I think that's really for a future date to get a better understanding. And sorry, Methven, I haven't left you too much time to go through the OS.
It should be quick. Yeah, it shouldn't be too bad. Yeah, we just have 2 minutes. But thanks. If there's no other questions,
I have a comment. Sorry, a question to Peter Hoff. So yeah, I'm gonna open a can of worms. That was like, yeah. Did any of the member of SAB raise the question of the selectivity against ARC5? The reason I'm asking that, because I'm not sure if we need to be selective or have dual inhibitor. because we have now 3 compounds or 4 compounds into the clinic against ALK5, and they have therapeutic window. And against, as you all know, against TGF beta, it's a very good immuno-oncology targets. So I don't know if someone raised that question.
Right. So in the SAB meeting, this really didn't come up, but we actually had some subsequent discussions that I don't have time to get into here with other folks about selectivity ALK2 and ALK5, and we can take that offline at another time.
Okay.
Mm-hmm. Yeah. Okay. If there are no other questions, let me just finish up the meeting with some of the comments from the CTIP. So like 5 days later, we had to meet with the OICR CTIP. Oops. So stop sharing. Then my presentation. Where is my presentation? Yep. Okay. Yep. So like as I say, we met on the 27th. We presented to them, gave them an update of the project, and so again, just to remind the group of what the objective of the project was 2 years ago, was to develop an ALK2 inhibitor for DIPG. And so these were some of the comments and feedback they had. So one of the first things is that they acknowledge that there is not a lot of good animal models for DIPG out there, other than some of the experts we're working with, the likes of Chris Jones and Angel and Nader. So those groups are building animal models. But they remark that the National Cancer Institute Pediatric Clinical Oncologist Consortium, they have this consortium that has been developed, and apparently they have animal models for childhood cancer. So they have invited us to really go and discuss with them evaluation of some of our lead compounds in these animal models. And as you're going to see later on, it's the NIH is one of the institutions we're actually now discussing with the next phase of the clinical development. They might get involved, and so they're really looking for good candidates moving forward. So the TPAC really acknowledged that. Also, the prioritized lead candidate 2163. I mean, one of the TPAC members asked whether or not the fluorine atom in 2163, resulted in PGP substrate issues. I didn't know where the line of that thought came from, but we mentioned to him that there were no PGB liabilities with that fluorinated dimethoxy compound. The TPAC also acknowledged that because of COVID, there was downtime. I mean, they had given us an extension up until June, but with the COVID, they give us more time to kind of complete all these in vivo efficacy studies. So they extended the timeline out until September 2020, so we don't have to rush. And so we were grateful for that. Like Peter just mentioned, there was a discussion of monotherapy versus combination. And like everybody thinks, similar, TPAC believes that, potential efficacy of ALK2 inhibition as a monotherapy, okay, owing to observation that ALK2 inhibition only results in tumor growth delay with some of the early data we had. They're convinced that combination therapy is going to be necessary, but they did support the team proceeding to the clinic as a single agent. I think that actually is essential as a first step. Okay, but they think later on single agent activity and combination therapy is also going to be things to consider going forward. Another point brought up again, just like Ahmed mentioned, I mean, in the beginning, some of the criteria we had in place was selectivity over ALK5, but because of microenvironment around tumors and TGF beta and ALK5 actually might become a part of the therapy. So they're starting to ask us to consider combination in some of these cellular models with ALK5 inhibitors. Or maybe some of our compounds that are potent at both targets might actually be good in therapy. Also, another point, because the standard of care right now is radiation therapy, the question came up about looking at combination of radiation with ALK2 inhibition. So that's something we're encouraged to start considering. And one of the things to keep in mind, we have a fall report due, one of the last report to the city that's due in October, end of September, October. And so they really want us to emphasize some of the clinical development plans. And like Peter just summarized some of the minutes, they want us to provide that in a summary report to them. So, and then finally, they gave kudos to the team. I mean, they feel like the project is a high-risk project, but those are the kind of things that researchers are encouraged to go after. And so the TIPAC really commended the team for what we have done so far. And they like the idea of us reaching out to groups like the NIH and stuff to secure additional funding going forward. But all in all, actually, they were quite excited with the progress for the project. And so these were some of the general comments. If there's any burning questions, the floor is open.
Method 0.5, do they mean like cellular models, like co-culture models with PBMCs and something like that?
Repeat that, PB. Yep. Yep. So there's
PMBC, whatever.
PMBCs.
Yeah.
I mean, they were just encouraging us to just look at combinations, and ALK5 was one of the targets. They never really specify which type of cellular models. So if people have ideas of some of the known co-culture models, might be one that we can also explore. I agree. Yep. Any other questions?
Rima, I don't know if you're on the line. I think I saw you earlier. I was just curious if you had any overall feedback. You're fairly close to the group. If there was any other color that you might have on this.
Hi, I'm on. No, not any additional feedback.
Okay, that's great. Sounds like they were quite pleased with the work, and I think they should be. I think this project has done quite a lot in 2 years. So, hats off to everyone who's on the line, who's put a lot of time and effort and support for this. It's been remarkable.
Yep, and again, I want to echo the same thing. Thanks to everyone for an excellent update. It had been a while, so there's quite a bit of update, a lot to think about. So we're going to have some discussions offline about some next steps, particularly with regards to some of the in vivo models. So again, thanks again, everyone, for hanging in there. We actually took it up to the minute, so I'm real happy, but we got to most of what we wanted to today. So thanks again, everyone. And
if I could just land one thing, I know a lot of people are traveling on vacations in August, to the extent you can go anywhere in August. But we'll keep you informed, but most likely the next meeting would probably be in September, early September. We'll keep you posted.
Okay. Thanks again, everyone.